Thank you very much, Perry, for that introduction. Just like to thank the organisers for the invitation to speak today. And might be a little bit of an unusual topic for this audience, but hopefully I've pitched the talk at the right level. So please bear with me. Can you see the correct screen there, presentation screen?
Yes, we can. All good. All
good. Excellent. So I'd just like to sort of run through about 4 different examples where molecular biology and genomics has been used in entomology, and just to give you some kind of sense for how those different techniques have actually shed light on various aspects of entomology, or we've able to taken things that we've developed through an understanding of insects and mechanisms of olfaction, etc., that insects use, and then apply them in the real world. Okay, so firstly, I want to talk about insect biodiversity and ecology, and there's no need to tell this audience that this is a challenging topic, and that molecular biology has been a useful tool in assessing biodiversity and asking very straightforward ecological questions about insects. But if we have a look at biodiversity here in the slide, you can see it's pretty straightforward to look at plants and vertebrates and assess biodiversity and their ecology. And certainly molecular biology tools have been really, really useful in opening up that ecological dark matter in those microbes and fungal and other very small organisms. But perhaps the area where it's been least applied and least thought about is in that kind of middle range of size where the insects sit. So I want to talk to you about one study that we've been worked with a number of years ago now on an island off New Zealand called Haturu, Little Barrier Island, where we've been applying these techniques to a whole range of biodiversity, including invertebrates and insects. This island's very interesting. It's the oldest nature reserve in New Zealand. It's as close to pristine as you'll get close to a large city. It's an extinct volcano, and it's very, very difficult to get to. And one of the reasons I was very interested in being involved in this study was actually to get to Haturu, because it's one of the last places in New Zealand where you actually have a full dawn chorus with all the birds and everything. We conducted a classical kind of ecological study where we did a classical approach sampling with large quadrats, 20 by 20 quadrats, to understand biodiversity, initially along an altitudinal gradient, but then more widely across this island, and use molecular barcoding and bioinformatics to assess biodiversity. We not only looked at invertebrates through leaf litter and pitfall traps, but we looked at all the plants, birds on these in these quadrats as well. Right, straight into the results. We sequenced not only invertebrates from leaf litter and pitfall traps, but we also took soil samples where we could look at microbes, including bacteria, fungi, using 16S ITS, and then also looked at invertebrates in the soil using cytochrome oxidase 1, CO1. And you can see here that, you know, across a huge range of taxa, that we're able to identify a huge range of different taxa types. And this has all been published now in GigaScience some years ago. We also use classical morphology to identify a number of the invertebrates in the pitfall traps and leaf litter, and sort of conducted a parallel study to the molecular study, which used next generation sequencing technology to sort of compare and contrast a classical approach to a sort of next generation sequencing metabarcoding approach. The data is incredibly rich and huge. Here's just some of we used Sanger sequencing of the vertebrates using cytochrome oxidase 1, and you can get a sense for the rich biodiversity here. Unfortunately, while we'd like to think that we know a lot about insect biodiversity in New Zealand, unfortunately, a lot of the species are undescribed. And in fact, the most commonly, or the most abundant invertebrate in our sample collection was a fungal beetle that's undescribed. Using this sort of phylogenetic approach, you can then overlay different lenses on it. Here we've looked at elevation to see if that is any pattern around elevation. You might be able to see there are some patterns there, and also the different trap catch types here, comparing pitfall trap and leaf litter. So these are very rich and powerful data sets. We can then go on and analyze data in different ways. Here we've used clustering analysis. First, we've condensed the data into OTUs, operational taxonomic units, based on sequence similarity, and then had a look at patterns both across the plots up the altitudinal gradient, and also clustering across the taxa as well. And then we can start not only looking at things like for diversity, but also look for other diversity measures, comparing beta diversity across the altitudinal gradient, which we've done here. Here we've compared, if I just get my pointer going, here we've compared some of the classical ways of estimating biodiversity in birds, trees, saplings, seedlings, and invertebrates using our CO1 sequence data, Sanger sequence data, with next generation sequencing from the soil. And we can see that we're uncovering much more biodiversity here when we are looking at this next generation sequencing metabarcoding data. And we can plot that data across the altitudinal gradients and as well looking by type. We couldn't actually see any pattern here, but it just illustrates the way that we can assess these data. One thing we did do, as a bit of fun, the suggestion of Alexi Drummond, was to use mark recapture as a way of comparing our data sets. So we've got our next generation sequencing data and our Sanger sequencing of individual insects, so next generation from the soil. You can see there's very little match between the two. One's from leaf litter and pitfall trap and the other from soil. So you might expect that, but using a mark recapture with these two different data sets, we can then gain estimates of biodiversity in these different groups on the island. And actually they're not far from estimates using classical techniques at all, which was in some ways kind of surprising that this approach would actually be useful at all. So if you're interested in this approach, please refer to the publication. So that's the end of that section. I think there's probably no surprises in there, but just to acknowledge that there's a huge effort's gone into this work. So I'd just like to acknowledge all my colleagues there, and this was done under the auspices of the Alan Wilson Centre, which is now defunct, unfortunately. Okay, so let's move on and have a look at how we can use these molecular biology and genomics techniques in say, looking at insect evolution and the origin of key innovations in insects. And yes, I've been interested in insect olfaction for a number of years now. So just a quick introduction. So the major structures that insects use to smell are their antennae and less so their maxillary pulps. And the main unit of olfaction is actually these sensory hairs, called sensilla. These sensilla are hollow, filled with an aqueous solution, and odorants enter through pores in the sensilla and are transported down to receptors that are on the dendrites of these olfactory neurons. So the question is, what are these receptors that receive and sense volatile odors? And it turns out that insects have their own multigene family. odorant receptor multigene family that sense smells, and it's different from the receptors that we use. So for many years, we thought they were the same because both our receptors and the ones that insects use both have 7 transmembranes. That means they are proteins that transverse the membrane of a cell 7 times. But the key difference is that the insect ones are in the opposite orientation to ours, and in fact they're not G protein coupled receptors at all. They're actually ion channels, and they also have this unique complex where they have a co-receptor orco that couples with a ligand binding receptors that actually detects the ligands, and together they form a channel that acts as the reception system. This gene family is the largest gene family in insects. It's huge. So many Hymenoptera can have hundreds, if not thousands, of these odorant receptors in their genome. And certainly it seems to be a key innovation for insects. Their sense of smells becomes, certainly in the higher insects, incredibly important, and underpins many of their essential behaviors from reproduction through finding food. So very important for insects. Key question is, when did they arise? So here's some early work by Hugh Robertson that suggested these receptors arose from taste receptors, gustatory receptors, with the co-receptor here right at the base of the evolution of the other odorant receptors, the ORXs. So this early indication that these receptors evolved from taste receptors sometime in the evolution of insects that we don't really know. So we've been interested in working out when the origin of these smell receptors in insects was, and we took a genomics approach. It's now no longer that difficult to sequence a genome or a transcriptome of the antennae of insects. So you can actually do these studies, and even they're within the bounds of a PhD to look at one or a number of genomes. So let's have a look, and we've used native New Zealand organisms so that we can use these genomes for other studies as well. So if we have a look at crustaceans, this is Daphnia here, one of the first crustaceans to be sequenced. There are no odorant receptors within the genome. They use other receptors. They can smell, and they use other receptors to do it, but they have a very small repertoire of these other receptors. And certainly there are none of these insect odorant receptor type receptors in their genome. So we sequenced Collembola. This is actually a Collembola collected off Haturu, Little Barrier Island. This is a giant Collembola. It's almost a centimetre long. It's almost frightening. It's so large. And this had a very small genome relative to other genomes. But within it, we found no odorant receptor genes at all. Only gustatory receptors and this other family of ionotropic receptors that primitive invertebrates used to detect some non-volatiles. We went on and sequenced a number of other native insects, including stick insects, where we did find odorant receptors. And here's the one of the largest insects by weight. This is the giant weta. This is also found on Haturu Island, right next to the major headquarters of the ranger, the dock ranger there. And this is quite a scary looking animal, and it also has a scarily large genome as well, which makes this very difficult to work with. But this creature also has a very large family of odorant receptors. Sequenced the giant bush dragonfly, perhaps more basal in the phylogeny of insects, one of the first flighted insects. We found in this, the genome of this organism, an orco and 4 ORXs or tuning receptors. So it has a very small repertoire of these odorant receptors. And of course, as you'll know, that dragonflies are quite visually orientated. So it's interesting to know, or to ask the question whether they had a low number because they don't use olfaction, or whether this is just we've captured something early in the evolution of olfaction with insects. This has been a PhD project of another student to look at the genome of mayfly, in this case spiny-gilled mayfly, native New Zealand mayfly. This has a reasonable number of odorant receptors in its genome, including an orco as well. And this is the work of a postdoc from Germany and collaborating with Bill Hansen's lab at the Max Planck, and where we've been sequencing more basal invertebrates, in this case silverfish and firebrate here. There are, again, these genomes of these organisms do have odorant receptors in them, and for one of them we've found orco today so far. So if we summarize all of that data, we can see that that odorant receptors evolved reasonably early in insects. And if we fill in with other data from other research groups as well, the bristletales, there's one bristletale sequenced, and in that genome there are a number of odorant receptors, but no orco has been found. And then if we fill in here with the silverfish, and then the dragonflies, mayflies, they all seem to have an orco plus these odorant receptors. So it seems that odorant receptors have evolved early in the hexapoda after terrestrialization, and then subsequently orco, the co-receptor, evolved after the origin of the odorant receptors, but before the evolution of flight, and maybe was required in the evolution of flight, because flying insects use olfaction as one of their major ways of navigating within that environment. Okay, so now this is the latest kind of phylogenetic tree of these odorant receptors. So still, we agree with Hugh that they probably evolved from gustatory receptors, but you can see around the base of the tree, we've got a much more complex situation with many of these more basal invertebrates having radiations of odorant receptors, including those related to orco. And then in the higher insects, you've got the more exclusive use of these ORX coupling to the orco. So there may be kind of different kinds of uses of these odorant receptors early in evolution, before this orco plus ORs system became established in the higher insects. Okay, I'll leave that there and now move on to a couple of other applications very quickly. So we've been interested in looking to see whether if we could apply our understanding of these receptors used for odor detection in insects to pest control. A lot of lepidopteran pests use a particular sex pheromone to find their mates. So females will produce or call, producing a pheromone which the males detect. And many applications in pest control are increasingly using either monitoring tools that use these sex pheromones to trap and see if the pests are around, or increasingly to disrupt this system in a technique called mating disruption, to confuse the males. They can't find the females. If they can't do that, then there's no offspring, so they interfere with the whole reproduction system. And this has become quite a successful tool in pest control. This is an Australian immigrant, Epiphys postfitana, or the light brown apple moth, and it's become a pest in New Zealand's horticultural systems. This moth's quite invasive. It's moved to Hawaii, to the US, and more recently into Europe. So the males have lots of these long trichoid sensilla that are sensitive to the female sex pheromone, and the pheromone receptors of these insects are a subclass of the odorant receptors. So our approach has been, well, if we could isolate these receptors, if we could work out how to screen them, and maybe even for their ligands, and then work out what compounds might be able to block them, could we then take these compounds out into the field and see whether these blocking compounds could confuse the mate finding behavior, or the males, and so they were unable to find the females. So this is just a progress report. So far we've identified the odorant receptors from this pest. There's about 70 of them. And it's been a long time coming, but finally we've identified some of the pheromone receptors in this species. And the reason it took us so long to work this out was typically the pheromone receptors were found in this subclade here, but it turned out in this species, and increasingly in other species as well, there's also another subclass of odorant receptors over here that are involved in pheromone detection. So we've also developed a cell line system where we can put these receptors into cell lines, overexpress them with the orco, and also other possible members of the signal transduction system. And then, because these receptors are calcium channels, we can then add calcium sensitive dyes and then detect influxes of calcium on the addition of the correct smell or volatile. And I won't bore you with the pharmacology, but basically you can, this is just like looking at a classical drug target, where you can, if you find the right odorant, then you can create a dose response curve for it, and you can see how sensitive it is to the different compounds. So here are 3 receptors that are sensitive to different components of the moth's pheromone system. We can screen huge numbers of odorants in these cell line systems. They're very easy to replicate and do high throughput screening on 96 well plates, so you can collect lots of data. So these are agonists, so that means they open the channel, and we can screen huge numbers of them. And here we're actually starting to look at antagonists. So these are compounds that block the ability of the channel to open in the presence of an agonist. So we pre-soak with the antagonist and then ask whether that blocks the ability of the receptor to open. And if we could find compounds that could do this, they may be effective at blocking these channels out in the field. So you can see that we're starting to screen for potential antagonists here. Unfortunately, nothing is really looking that efficacious in these cell lines so far. But I think this, at least we've got proof of concept that this approach may well work to find candidates. And then finally, I just want to point to a group from the lab that has spun out into a company more recently, but through our understanding of how these receptors work, and also the ability to transduce the signal across into classical biosensing platforms. So your mobile phone can do a lot of things, but one thing it cannot do is smell. And if it could smell and taste, it would be very, very useful in a whole range of applications, straight from the food industry right through to sensing disease on the breath. So there's been quite a few groups very interested in seeing if we can miniaturize this task, either using artificial systems or using nature's own receptors, whether they be our smell receptors or those of insects, which we know are very sensitive and can be very specific to a range of compounds. So we've learned how to overexpress these receptors, solubilize them, and then put them into formats that are amenable to putting on biosensor surfaces. Here are 2 different formats, liposomes, which are spheres of, and then there's also nanodiscs as well. And these different, the receptors in these different formats have been placed onto different types of biosensor surfaces. Here we're using electrical impedance spectroscopy. for a couple of receptors. In this case, it's OR10A, which detects methyl salicylate, either with or without orco, showing the ability of it to bind its ligand. Methyl salicylate smells like liniment. And here's another receptor, OR22A in nanodiscs, detecting ethyl hexanoate. And you can see that the sensitivity levels are quite incredible, down well below pico femtomolar. So in these formats, these receptors are incredibly sensitive biosensors. And then finally, this is in collaboration with researchers at Victoria University, Wellington. This is using graphene FET. So here, the receptors have been, in this case, both formats again, liposomes and nanodiscs, and they've been attached onto graphene surfaces and then used as biosensors. And again, with the two receptors 10A and 22A, both shown to be very, very similar, sensitive with limits of detection down at around 1 femtomolar. So that's very, very sensitive. So as said before, this technology has been patented and spun out into a startup company called Sentient Bio. And the company spends a lot of its time mass producing insect odorant receptors and putting them on these biosensor surfaces, and developing partnerships with biosensor companies to actually make olfactory sensing elements. So there's many people to thank with all of this section of work, from talented PhD students, collaborators such as Thomas Buckley, who I've worked with on the insect genomics area, with Jacob Corcoran and Melissa Jordan on the pest control side, and Andrew Krylacek, who was a postdoc in the lab originally, is now the CTO of the startup company Sentient Bio. Okay, so just to summarize, hopefully I've persuaded you that metabarcoding is a useful tool for looking at insect biodiversity assessment and ecology, that using genome sequencing now, we can actually start to look at some of the key innovations and the origins of gene families in insects. And that through looking at some of these gene families of odorant receptors, we may be able to start thinking about novel pest control applications and also applications in biosensor development. Okay, ngā mihi nui, thank you very much for your attention.
Thank you, Richard. That was a very interesting talk. So we are actually running ahead of time a little bit. We do have time for questions. So if you'd like to place any you have in the chat, please feel free to. Okay, Richard, it looks like we do have one in the chat from Ari Hoffman. Would you like me to read that out to you, or are you happy to speak to that yourself?
I can read that. If I can flip the issue around, where has genomics not delivered? Oh, that's a great provocation. I think where it's perhaps not delivered is I think that we probably don't have as many tools as we'd like to do what you might call functional genomics, to understand the role of those genes. We have crude techniques. We've obviously got CRISPR now that we can use, but I think that we're still, while we can collect information about what genes are in a genome, I think we're still very limited in terms of moving to sort of functional understanding and looking at how different genes are contributing to different pathways, processes within insects. So I think that's still going to be quite slow.
Yeah, thanks, Richard.
Martin has had his hand up for a while, so I think we might take one of his questions next and then move back to the chat.
Fantastic talk, thanks, Richard. I'm just curious, what's the relative benefit of an electrophysiological approach to identifying you know, key odorants over a molecular approach?
Yeah, so there are a couple of different ways of doing this. I think, I mean, obviously with electrophysiological approach, you don't have to go through that whole molecular route, and you're probably making less assumptions. You're just asking the question, given this compound, can I detect a depolarization event going on? So both electrophysiology and molecular approaches suffer from the same problem, in that they don't tell you anything about the polarity of that response in terms of how it's translated into a behavior, whether it's a repulsion or attraction, or I don't really care. I mean, that's probably as a third as well.
Yeah. Thanks very much. Yeah.
Okay, so we've got 2 more questions in the chat at the moment.
Good to hear from you, Wei. Could we use gustatory receptors in your biosensors? Quite possibly, yes. In fact, the formats that we're using are still aqueous anyway, so we're still using these odorant receptors in an aqueous environment rather than in a volatile environment. So yes, they may well work. And there's another question there from Kirsten Ellis. Is there a potential future use for metabarcoding with insect monitoring in crops? Absolutely, and I think as we've seen with COVID, some of these rapid sequencing technologies that can go out into the field, we may well start to see these technologies be even deployed out as monitoring tools.
Okay, fantastic. So it doesn't look like we have any more questions at the moment, and we are about 10 minutes ahead of time.